Hello everybody, welcome to the ICSR series, the most demanded and awaited video for all the freshers and experienced people who are looking to improve their knowledge and trying to crack the interviews. I have received numerous requests from so many candidates and I have put in all the efforts to make it as simple as possible and easy to understand the concepts. The ICSR case processing is very important, which makes the foundation and also the entry point in the drug safety and pharmacovigilance domain. If you understand and master this process very well, you will be able to advance and shape your career into aggregate reporting and so on. The ICSR case processing provides great potential and immense opportunities for freshers to start their career because most of the big pharmaceutical companies and biotech companies generally outsource this activity as these projects usually require huge resources to deal with large volumes of work. And also, many organizations have to deal with the challenges of quality and productivity, mainly due to the lack of proper training and concepts being not clear. So let's see in detail what is pharmacovigilance, definitions and terminologies used, and where do we get the data, how it is collected, documented, processed and then reported to the regulatory authorities. What are the tools and the safety databases used? As many of you already know by now, my main focus is always on the practical aspects rather than just learning some theory part. My intention is to give you tons of information on all my videos. I think that I have covered a lot of important topics on my videos, which I feel is more than sufficient for you to ace the interviews. Please watch all my videos till the end to get a proper and thorough understanding and comment if you come across any questions. Also, at the end of each video, I have included a small mock interview session where I will be asking you the questions and you can try to answer, which will help you practice your flow and improve your speaking skills. One more very important thing, if you are still in college or pursuing any of the streams in medicine, dentistry, pharmacy, PhD or any other life sciences degree and not sure of practicing and would like to know more about the career path and options in the pharma industry and you are in need of guidance, you can ask your college authorities or the concerned person to contact us by email or send an invite for a session to talk on pharmacovigilance and clinical research and the opportunities in the industry. Of course, the session will be entirely free and I will be happy to see you all and talk about in length about pharmacovigilance and clinical research. I hope you will enjoy all my videos. Hi everybody, welcome to the Causality Assessment in Drug Safety and Pharmacovigilance. In this video, we will be looking at Causality Assessment methods in detail. The table of contents, introduction, background, why causality assessment, what is the need for causality assessment, principles and methods as well as the conclusion. I have referred to two websites, that is National Library of Medicine as well as WHO-UMC org website. Let's look at the introduction. Causality assessment essentially means finding a causal association or relationship between a drug and drug reaction. Please remember this. Identifying the culprit drug or drugs can be life-saving or helpful in preventing the further damage caused by the drug to our body systems. There are multiple criteria or algorithms available as of now for establishing a causal relationship in cases of adverse drug reaction, indicating that none of them is specific or complete. This is an important and challenging part of pharmacovigilance in which the attempts are made to find out the exact drug responsible for causing drug reaction. This is very important in clinical practice as more and more drugs are flooding the market and are used by our patients and more likely to cause side effects beside its effects. In the pursuit of efficacy, safety of these drugs is usually ignored. As the safety of the patients is more important than efficacy, identifying the culprit drugs become much more essential. The principles and methods of causality assessments or causality assessment tool help clinicians to identify the culprit drugs. Please note the causality assessment tool, used commonly abbreviated as CAT, in my next upcoming slides, so that you don't get confused with another synonym or any other term related to CAT. C-A-T. Just a bit of background. Healthcare industry is flooded with multitude of drugs and the list is increasing day by day. Consumption of medications has enormously increased due to lifestyle changes, and having safer drugs is the need of the hour. To have a check, regulators and other authorities have put in stringent regulations and pharmacovigilance system in place. Even though there has been increase in adverse drug reactions reporting in the last decade, causality assessment has been the greater challenge for academicians and even industry. Causality is crucial for risk-benefit assessment, particularly when it involves post-marketing safety signals. Pharmaceutical companies have put in efforts to have a standardized approach for causality assessment. So now, why causality assessment? An inherent problem in pharmacovigilance is that most case reports concern suspected adverse drug reactions. Adverse reactions are rarely specific for the drug. Diagnostic tests are usually absent and a re-challenge is rarely ethically justified. In practice, few adverse reactions are certain or unlikely. Most are somewhere in between these extremes, that is possible or probable. In an attempt to solve this problem, many systems have been developed for a structured and harmonized assessment of causality. None of these systems, however, have been shown to produce a precise and reliable quantitative estimation of relationship likelihood. Nevertheless, the causality assessment has become a common routine procedure in pharmacovigilance. The advances and limitations of causality assessment are reviewed in the below table. Please have a look. Need for causality assessment. Causality assessment is very important in every discipline of medical practice. Identifying the culprit drug or drugs can be life-saving or helpful in preventing the further damage caused by the drug to our body systems. In practice, this is much more important and relevant because many etiologies can produce a similar manifestations. Many a time, multiple drugs are taken by the patient and it is difficult to ascertain the exact drug responsible for causing a drug reaction, and when the underlying disease is capable of producing the same manifestation as caused by the drug. Unfortunately, there are no specific diagnostic tests. Although histopathology can provide a clue for the diagnosis of drug reaction, it is not sufficient to point toward a particular drug. However, identifying the exact drug is of paramount importance as it directly influences decision making by the clinician either to continue or to withdraw the likely drugs. This can be very critical in some circumstances, and thus establishing causality with the near perfection is desirable. In such circumstances, we require the help of some time-tested CATs as outlined in this review. Causality assessment can be undertaken by clinicians, pharmaceutical industry people, academicians, and regulatory authorities in different settings, including clinical trials. Let's look at the principles of causality assessment. All causality assessment methods or tools follow four cardinal principles of diagnosis of ADR. So please remember these four things. The first one is the temporal relationship of drug with the drug reaction, biological plausibility, de-challenge and re-challenge. When de-challenge or re-challenge has occurred in the past, it is called positive pre-challenge or negative pre-challenge. Now let's look at what are the methods of causality assessment. Causality assessment methods are broadly classified into clinical judgment or global introspection, algorithms, and Bayesian or probabilistic methods. We have three. The total number of methods as included in the above classification is 34 methods based on the results of a systematic review. Needless to say, all these methods require inputs or information by a clinician, and thus they are useless unless a clinician gives his or her opinion on a given case. Now let's see the first one, that is clinical judgment or global introspection. As far as a clinician is concerned, clinical judgment obviously is helpful in identifying the relationship likelihood in actual clinical practice. Knowledge and experience of the clinician plays a very important role in performing global introspection. When a number of drugs or more, an underlying disease can produce a similar manifestation, clinical judgment or global introspection has limits. Furthermore, there are individual variations in causality assessment because of differences in knowledge and experience of clinicians. To impart some degree of uniformity, two methods have been suggested. These are Swedish method and World Health Organization-Uppsala Monitoring Center, that is WHO-UMC scale, which are clinical based and clinical judgment based CAT, causality assessment tool. Of these, the WHO-UMC scale, which is based on the knowledge of clinical pharmacology, is widely used in individual case assessment. So this can be an interview question. So WHO-UMC scale is the most widely used. The WHO-UMC table is given below. I will be showing you in the upcoming slide. Basic criteria for causality of these methods are knowledge about pharmacology and previous information of ADR, association of AE, that is the time, place, etc., and a drug, biological plausibility and exclusion of other causes. This is the World Health Organization Uppsala Monitoring Center causality assessment scale, that is we have certain, probable or likely, possible, unlikely, conditional or unclassified, unassessable or unclassifiable. So depending on the assessment criteria, you assess the causality term, whether it is certain, probable or likely or possible or unlikely. Please go through this. I will give you some time to go through this. Coming to the second one, that is the algorithmic methods. Algorithms are simple and mostly questionnaire based CAT, in which scores are assigned to answers of its questions, and the given ADR is categorized into categories such as definite, probable, possible or unlikely. Some of the most commonly available criteria used across the globe are Naranjo's algorithm, Kramer algorithm, Jones, Karch, Begod algorithm, adverse drug reactions, advisory committee guidelines, etc. Many of them are also modified. You can Google each one of them to find more information on these methods. Algorithms are inflexible and have been designed to reduce intra-rater and inter-rater variability to make this CAT more reproducible and valid. The details of the Naranjo scales or algorithm are given in the table 2. I am showing you now. So here we have questionnaire. We ask question, are there previous conclusive reports on this reaction? So if it is yes, we rate it as +1. If it is no, it is 0. If it is don't know or not done, then it will be considered 0. You can see all of them are 0. If it is don't know, you put it as 0. So you use this questionnaire and you use all this question and you come to a score. When the question in the above questionnaire are answered by a clinician, a score is obtained. Based on this score, ADR is categorized into the following 4 categories. So this will be asked in the interview, and I have also added this question at the end of the interview. So please remember this. If it is more than or equal to 9, then it is definite ADR. If it is within the range from 5 to 8, then it is a probable ADR. If it is between 1 to 4, then it is possible ADR. And if it is 0, then doubtful ADR. So 9, 5 to 8, 1 to 4 and 0. Please note this, and when asked in the interview, just mention definite, probable, possible or doubtful, only 4. Coming to the Bayesian or probabilistic methods. Bayesian methods follow different and complex statistical approach for finding the cause of a drug reaction. It includes Bayesian Adverse Reaction Diagnostic Instrument, abbreviated as BARDI. This considers prior probability of the drug in question as obtained by prior epidemiological studies, combining it with the likelihood ratio as obtained by a given case information. In short, it calculates so-called posterior probability or posterior odds of the drug causing a given drug reaction. Prior odds factor is the ratio of expected drug attributable risk to the background risk of a certain AE. The likelihood ratio is calculated for history, timing of the AE with regard to drug, characteristics of the event, de-challenge, referring to signs and symptoms after drug withdrawal and re-challenge. Bayesian methods use Bayesian spreadsheets, which are complex and cumbersome for a clinician as it involves complex statistical calculation. These are now converted to easy computer-aided programs such as MAC, BARDI, Q and A, question and answer. Thus, given our limitation about understanding the knowledge of CADR and drugs and lack of objective diagnostic tests for the diagnosis of CADR, CATs as outlined above should be used in clinical practice, accepting their pros and cons. There is no substitute for clinical observation and experience of the clinicians when it comes to identify the cause of drug reaction. Safety conscious physicians can help in reducing mortality and morbidity associated with ADRs using their clinical acumen assisted by some CATs. Coming to the conclusions, the end of the slide. In pharmaceutical industry, structured causality assessment methods for individual cases have been used to facilitate case processing rather than to reach definitive conclusions. Events are considered related or not related for the purpose of case processing and regulatory reporting. Definitive decisions about causality leading to risk management plan or label changes, etc. usually incorporate evidence from various sources. However, no single one-size-fits-all approach exists, as sometimes individual case reports provide compelling evidence of causality. So overall summary, it is up to you. So we have the 4 principles and 3 methods of causality assessment. The most widely used is the WHO-UMC scale, and there are other methods, but we do not have any one size fits all, or we do not have a standardized scale which will give you the accurate information about the causality. Now the time for interview questions. What is a causality assessment? What is a temporal relationship? What are the methods of causality assessment? What are the terms used for causality? Which is the most widely used CAT? How are ADRs categorized based on the scores in Naranjo Scale? What is the causality assessment for spontaneous reporting? So if you have not watched my previous one of the video in the ICSRs for spontaneous reporting, please watch it. The answer is in that video. Hope you were able to answer all these questions. So I am conducting a training session on aggregate or periodic reports, which will be on DSUR, PSUR, PBRER, signal detection and management, as well as RMP and medical literature or literature surveillance. These sessions will be conducted on weekends from 5:30 or 5 p.m. to 8:30 p.m. IST, usually on Saturdays and Sundays. So if you are interested, you can visit our website www.globalplacosolutions.com. Under contact us, you can navigate to the training, and under that you have option, you can select the training option and you can register yourself. If you are interested to attend any of the session, we will be, after the registration, we will be sending you an email with the available schedule on the session along with the fees or the charges. You can attend depending on your convenience. I hope that the video was informative and helpful for freshers, especially if they have not heard about the causality assessment as a new concept, and those who are preparing for the interviews. I would like to thank you all for watching and wish you the best of luck for the interviews.